structure is hollow tubes. Wall consists of 13 columns of tubulin molecules. So basically these tubulin molecules will arrange themselves, 13 tubulin molecules like that arrange themselves, and that way it's going to form a hollow cylinder structure, and that gives the strength. Diameter 25 nanometer with 15 nanometer lumen. The made up protein is tubulin, and main functions: maintenance of cell shape, motility of a cell, chromosome movement during cell division, and organelle movements inside the cell. Because these microtubules will form path inside of the cell, inside of a cell, so that organelles and other cargo proteins can be transported. Just like there are motor proteins who has this cargo on the top and they move and walk like that. It's a molecular walk, resembling molecular walk on the surface of microtubule path. That's why we are calling it as helping in the organelle movement or cargo movement. So let's first start talking about microtubule filament. Microtubule structures. We don't call it filament because it's thicker, the thickest among the cytoskeleton. So what is microtubule and what is the structure of microtubule? You can clearly see plenty of pictures provided here. Let me give you a simple idea. We have already talked about the basic constituent of microtubule. We call it, it's made up with tubulin, remember, tubulin protein, and basically made up with tubulin dimer. There are two types of tubulin, alpha tubulin and beta tubulin, beta and alpha. Beta is denoted with red color, alpha is denoted with blue color here. So beta and alpha tubulin together will form tubulin dimer, and the tubulin dimer will be in contact with one another top to bottom. You can see the contact is top to bottom, beta on top, alpha on bottom. That way top to bottom arrangement is there, while they make a top view of 13 different microtubule, 13 different tubulin proteins together. So basically 13 protofilament is a typical cylinder. You can see that diameter of 23 nanometer, and this 13 different tubulin they arrange together. So basically they first arrange themselves like a sheet. They form a sheet like structure like this, and then they'll fold the sheet to form a cylinder. So it's forming a hollow cylinder, okay, and there is a plus end which is fast growing end, and there is a minus end, slow growing end, okay. The fast end or plus end with beta tubulin at its end, and the slow growing end has a alpha tubulin at its end.